Learning objectives include antibiotic sensitivity testing, minimal inhibitory concentration, and is the drug bactericidal or bacteriostatic? Well, physicians usually prescribe drugs that they know that those drugs will work. But that is not the case always. Sometimes infections are caused by such an organism that are resistant, or the drug that the physician prescribed does not work, and infections remains there. So in such cases, if the organism has been isolated and identified, we can test the organism for sensitivity to various antimicrobials. There are various ways of testing for the sensitivity. One of them is called disk diffusion method. In disk diffusion method, an inoculum of the microorganism, which has already been identified, is spread onto the agar plate, and then filter paper discs, circular filter paper discs that are soaked in some concentration of these various antibacterials, they're placed over the agar plate, and then the whole thing is incubated in the incubator for like overnight. And next day, we read the results. This is a picture that shows that. As you can see, that these white discs, they are paper discs that have been soaked in various antibiotics. As you can see that around the antibiotics, you don't see the growth of the organism. And this area is measured, the zone of inhibition. This area, clear area around the disc, is called zone of inhibition. So this is measured, and then this measurement is then compared with a standard table. So companies, when they supply these various discs for various antibiotics, they also provide their table, which has been worked out that if this much is the zone of inhibition, it means that the bacteria is sensitive, or if this is less than that, it is intermediate, or if it is not, then it's resistant. So each disc is then recorded as sensitive or intermediate in its sensitivity or resistant. As you can see on this plate, there are certain areas that are larger, and that indicates that if you use that antibiotic on the patient, the organism will be killed. And you can see at least there are 2 discs that do not show any zone of inhibition. They are resistant. So if you use those antibiotics for some chance, bacteria is not going to go. And then there are zones that are intermediate in their zones of inhibition. This is a little modification of the same test that we used previously. Here, you can see that a long strip, instead of a circular disc, a long strip of filter paper is used. Here, sometimes the physician wants to know minimum inhibitory concentration. So how this is calculated or how this is done is so easy. As you can see that these strips, they have a concentration gradient of that antibacterial. So on one side, as you can see, the bottom side is each of these strips. They are different antibiotics. And on one side or one end of the strip is low concentration of the antibiotic. And as you go above or upward, the concentration of antibiotic changes. It increases. And as you can see, that as the concentration of the antibiotic increases, so does the zone of inhibition. So minimal inhibitory concentration can be read from the strip, where you see that at that concentration, the zone of inhibition start appearing. And this gives an idea to the doctor or the physician that how much concentration the antibiotic would be given, because although on the strip there may be higher concentration available, but not necessarily the physician would give that concentration, because if that drug is the only drug available, but at a higher concentration it is toxic, so doctor needs to know how much is the minimum inhibitory concentration and what is the maximum. So he would stay somewhere in the middle. There is another drug testing procedure that is called broth dilution method. In this method, various concentration of broth are made with the same antibiotic. You can make 2-fold, 10-fold, or whatever. And bacterium that has to be tested is inoculated into those broth tubes or wells. It's like this. These are micro wells of a plate that has various dilution of the same antibiotic. In these 2 rows, you can have 2 different antibiotics. And as you can see, that white fluffy part is indicative of bacterial growth. And if you start reading from the left to the right, you see no growth in the bottom 2 wells. The third well is showing a growth there. So here, the minimum inhibitory concentration is the second well. And the beauty of this broth dilution test is that you can test whether the antibiotic is bactericidal or bacteristatic. If you take one higher concentration than the minimum inhibitory concentration, like in this case, well number 1, if you take the cells, bacterial cells that are there, and inoculate those cells in another plate, a fresh plate of Newton agar, and incubate it overnight, and if you see the growth there, okay, there could be 2 scenarios. There is no growth or there is growth. After overnight incubation, if the plate shows growth, what does it tell you? It tells you that the microbes, they were lying there intact but were not replicating. So that means that the drug had a bacteriostatic effect. But if you don't see a growth in the plate that you inoculated with number 1 well here, at that concentration, that means that the cells, they were not able to make any colonies. They were not able to grow. So that means that the drug was bactericidal. It lysed the cells, and there were no more intact cells there. So this broth dilution test is a great way to know whether an antibacterial is bactericidal or bacteristatic. So this is the same thing that I just described. There is a third concept. It's called antibiograms. Antibiograms are basically profiles of various drugs. All physicians in their area, they know that what kind of antibiotic is working these days. And the key is basically that you isolate the organism from the patient and you run a battery or a host of these antibiotic sensitivity testing, and you know that Staphylococcus aureus is sensitive to these antibiotics and is resistant to these antibiotics, and this is called antibiogram. Utility of antibiogram could be that if a doctor knows that these antibiotics are no more working, so he doesn't have to waste time and money on prescribing those medicines. So in summary, there are 2 different ways of testing the sensitivity. One is disc diffusion, and the other is broth dilution method. Broth dilution method has advantages of telling you whether the drug was bactericidal or bacteristatic. And one can create antibiogram or antibiotic profile of an organism that can help a clinician or a physician to prescribe the right medicine. Thank you.